Good, good, good. Now we're going to talk about all the rest. Time check. Need to get a better timer. Ooh, maybe I just put a timer on here. We're at 10 minutes. We're good. Let's go through all the rest, and then we will be half done with lecture for this unit. Wow. So this last part is just all the rest of the organelles. They're largely like structural, skeletal things. They're less important, but still will come up. So first, the cytoskeleton. An often neglected and not shown part of cell models is the cytoskeleton. Because if we did show it, it would just make everything really confusing. But essentially, there's a bunch of, think like collagen fibers almost, that extend throughout your cell, and it gives everything structure. It like holds the mitochondria in place. It creates these like highways and pathways for vesicles to travel along. It helps the cell move itself. It just kind of, it gives everything structure. It's like the road system of inside the cell. Without it, everything would happen much less efficiently, because everything can kind of just walk along the tracks. The tracks are going to be more efficient than just randomly traveling through the cytoplasm. Made up of 3 main fibers. Really don't stress about these. I wouldn't even bother to define them and write them down. They're tubules, microfilaments, and intermediate filaments. It used to be more emphasized in the AP exam. I haven't seen it show up, especially with the specific differences. But generally, you have your actin filaments, the microfilaments. These help maintain cell shape. These do muscle contraction along with myosin. They are involved in cell division and moving organelles around. Then you have your intermediate filaments. These are keratin, which is the main protein in your hair and in horns. These also maintain cell shape and help to anchor some things, just kind of like a bigger steel cable. And then you have your microtubules, which are the biggest ones. These move the whole cell. They move chromosomes, provide tracks. They're kind of the biggest level of it. But again, I wouldn't worry too much about them. Cilia and flagella, hard to say. They help cell move. Generally, the difference between them is just how big / how many do you have. So flagella are usually kind of one long one, like in the euglena shown there, or in a sperm cell. Cilia are a bunch of tiny little hairs, kind of like the legs of a centipede, that all move together to help move the cell around. But sometimes we will use cilia for other things. We have cilia on some of our cells in the human body, despite not really using it for movement. What we use it for is to help keep junk out of our lungs. So we have a mucus layer along our trachea entering our lungs, with a bunch of cilia kind of underneath it. So the mucus kind of lies on top of the cilia. And the idea is that as we breathe junk in, like campfire smoke, any kind of smoke, any debris, anything that's not oxygen and nitrogen, it'll get trapped in the mucus, and then the cilia will kind of wave it and move it and migrate it out away from the lungs, and usually you cough. If you've ever inhaled smoke at a campfire, this is what's happening. The smoke goes in, gets trapped by the mucus, brought back up, and you cough because you've just inhaled the smoke. Cell wall, we talked a little bit about. It's in plants and some single cell organisms. Maintains shape, prevents too much water from entering. Made of cellulose. In bacteria, that says it's made of peptidoglycan, which we already talked about, which is why I'm not worrying too much about my faulty slide there. Cell junctions. Essentially, our cells are not really free floating. They are in constant contact with their neighbors, other cells. And often this is done through cell junctions, where cells can basically talk with each other, exchange material back and forth, and stick together. In plant cells, the cell wall itself has a bunch of holes in it called plasmodesmata that again allows cytoplasm to be exchanged between the two cells freely. In animal cells, we have three main types. Don't worry about the distinct differences. Just know that animal cells have cell junctions. Don't worry about the distinct types. So essentially they can be either tightly pressed against each other, called a tight junction. They can function kind of like a rivet and link each other into sheets using keratin. We use this with muscle cells, can drag them with each other. And then gap junctions are more like a membranous pore kind of connects the two, makes a channel between the two that allows material to be shared between ion, sugars, amino acids. It's very important for communication. Really, in terms of this cell junction idea, really just think of it as cells are connected and they can send material back and forth to communicate. That's it. Don't worry about the names. Don't worry about the tiny specifics. As always, these, this all the rest section, probably the least important. I wouldn't focus too much on getting good notes in it. That is all of the organelles of the cell that we need to know. We will be doing our cell organelle dating game and then spending a ton of time talking about the cell membrane. Tons of fun. Let me know if you have questions. And as always, that's all. I'll spin my propeller. It doesn't spin well. Bye.